So hello everyone and welcome. Hopefully you can see and hear me okay. And now we slowly get started for the last lecture of the lecture series, which is the lecture series called This is Public Health, which is brought to you by ETH and the Swiss School of Public Health. For those of you who've been joining us, then great to have you back. And this is the last session of the series before the midterm break. So pleased that you could join us. As always, I normally get started by encouraging you to download the quiz on sustainable health. So if you haven't done so already, then I invite you now just to take this moment to scan this QR code and take a look at what the community of the Swiss School of Public Health has contributed to. It's an interesting quiz all about health in relationship to all sustainable development goals that was built by, the questions came from faculty members and from students from across all these different universities. This quiz was a strong feature at the Dubai Expo. So if you are not familiar with me already, then you may know me from the Dubai Expo project. My name is Emily Reeves. And I was previously managing the Dubai Expo project, and now I am moderating this session. So thanks for being here. I link again, just as always, highlighted in yellow, some services to counseling services or to support services. There are quite difficult and quite strange times that we live in at the moment. So if you are feeling somehow impacted in any way by any of the circumstances taking part in the world at the moment, then please don't hesitate to reach out to where you can find help through that link there. I thought I'd start this time just by mentioning, for those of you who have asked about the midterm quiz, I thought I would just reiterate the facts on that as it is now coming up. The quiz will in fact be posted on Moodle this upcoming Monday, and it will remain open for 2 weeks. So you have 2 weeks to complete it. Although once you do start the quiz, you have 3 hours to finish it. The quiz will be in English, and it will be based on 21 multiple choice questions, which are about the lectures that you received, and there'll only be one correct answer. If you are unable to complete this quiz to 60% pass rate, then you will have to do a written assignment instead. But you are absolutely allowed to look at your notes and reflect on the lecture material that is posted online. Everything is available in the program, so you can see all of the lecture slides, and you can even look back and watch the video recordings of the lectures as well, if you'd like to do that in order to be able to answer the questions. But of course, sharing the answers is not allowed. So that's for those of you who've asked about the quiz, and I will circulate again information about this this week, and I will also, of course, inform you when the Moodle is ready on Monday so that you can get started with that. So without further ado, then that brings me to the lecture today, which is monitoring COVID and combining, of course, clinical and wastewater surveillance. And this is brought to us today by Dr. Jana Huisman, who is a researcher at the Department of Environmental System Science at ETH in Zurich. So it's really a pleasure to have you with us here today, and we look forward to hearing more from you. So the floor is yours. Thank you, Dr. Huisman.
Thank you, Emily. Oh, I cannot share my screen yet. 1 second. So now you should be able to see this presentation. Yeah, as Emily said, I was planning to talk today about the monitoring of SARS-CoV-2 we've been carrying out over the past 2 years now. But in discussion with the organizers, part of my presentation today will also focus more generally on the role of mathematical modeling in public health, which questions we can answer with mathematical modeling, and which role it has been playing in policy decisions also during the SARS-CoV-2 epidemic. So probably all of you have encountered mathematics already during your studies, and specifically also in relation to public health. It comes in various ways. most prominently probably in statistics, when planning experiments or analyzing them and making predictions for the short-term future. And there's quite a bit of modeling already going into this aspect. Then there's mathematics that comes in sort of in the bioinformatics area. Nowadays, we collect a multitude of what we call omics data, genomics, proteomics, these kinds of data, and just to analyze these sheer volumes of data we can collect on study participants, for instance, or on viruses and bacteria as they infect different people and spread through a population. All of this data is analyzed using typically mathematical tools and what we call bioinformatics. But specifically what I want to talk about today is sort of mathematical modeling, really in the sense of constructing models that describe an aspect of reality and studying then those models in more detail, what they tell us about the underlying system. And there's different types of models that one can construct. Most broadly, typically sort of within host models, where we might describe, for instance, the way the body reacts to an infection with a particular pathogen, describing how the immune system starts to react to the pathogen, how, for instance, the kinetics of an antibiotic might occur over the span of multiple days as it's being degraded, and how that interacts with the pathogen within that system. Or also between host models, where we're essentially studying the spread of a disease through a population. This is sort of the primary focus that I've had over the past few years on modeling, first of all, the spread of antibiotic resistance through bacterial populations. It's these kind of between host models, but also modeling the spread of SARS-CoV-2 between human populations. Then one can incorporate many layers of additional information, for instance, about the behavior of the virus, the pathogen that is spreading, but also of the hosts that it's spreading between, some of the kinetics of spread and so on, and also ask questions to this model of what would happen if you were to intervene with certain aspects of the spread, for instance. In this paper that I shared as recommended reading, they have a wide array of examples of mathematical modeling in public health, and also sketch sort of this general a set of steps that are followed when trying to construct a mathematical model and answer policy relevant questions. So it typically starts with the question in the first place. Here in this figure, they suggest, should we introduce vaccination against a particular disease? And if so, what's the best strategy to introduce this vaccination? And once you have this question, you start to wonder about two things sort of in parallel. First of all, how can we model the question and try to answer it? So in this case, what's the disease that we're studying? Who are the people that might be infected by it? What are the dynamics of infection, and so on? And also, what data do we have available to inform our model and to test it? So first of all, to parameterize, for instance, the rate of transmission of the virus between different people, but also to see what is the current level of people infected with the virus, and if we were to introduce vaccination, can we test what that has done to our levels of virus incidence, for instance. And then once we start with these components, of course, we actually have to do the research. So once we have an idea of the model, we need to implement it and test it in different situations. We might ask more specific questions, for instance, of strategy. Should we target specifically old people or young people? Should we try to target everyone all at once or in cohorts over multiple years? And in the model, you can ask a multitude of these questions, and each time sort of rerun the model and check what the suggested outcomes might be. And with that, you can formulate a set of conclusions or advice to say, okay, our model has predicted that this is the optimal strategy to implement vaccination within this particular population, for instance. And we find that it might be susceptible or sensitive, our predictions might be sensitive to these parameters, for instance, the age structure of the population, more than to other parameters, such as the rate of recovery, perhaps, from an infection with this particular virus. And with that advice or this conclusion, then policymakers can combine it with other forms of information and advice and decide, okay, should we now implement, for instance, a trial of vaccination in a particular population? And with that, can we generate new data to test whether the predictions of our model made sense, yes or no? And then we sort of come back to the beginning and can start to refine our model or perhaps ask new questions from it once we know that it's a sensible way of describing the underlying dynamics of the virus in this case. And so perhaps you wonder, why would you want to do modeling in this case? Why not just and I think the key advantage of modeling can be to, first of all, sort of test many counterfactuals, many scenarios that you might not have the money to carry out tests for separately in all of these cases. Or that might be unfeasible simply because the population that you need is too large, or because it would be unethical not to give people the optimal treatment in a particular case. And with modeling, you can essentially explore a lot of these different outcomes and again formulate recommendations. What also can be done is sort of to test our own mechanistic understanding of a particular biological system. So essentially, if you can model it, this shows that you understand what's going on, because you need to come up with parameters and descriptions for each step in the process. And if you find that your model does not, or the predictions from your model do not correspond to the reality that you then observe when you carry out the experiment, or when the process evolves, you know that you need to update your model, and until you've basically captured all the essential components of what it is that you're trying to describe. And lastly, of course, what's really useful is that you can generate quantitative insight. You can predict X many people will be infected with this disease by this and this date, given the current circumstances. Or we expect this many deaths after 2 weeks, or we can save X amount of costs if we vaccinate in this manner versus that manner. And by generating this quantitative insight, you also give policymakers the tools to sort of evaluate one scenario versus the other, and really compare them in that sense. And I think here it's important to remember, to internalize this feeling that all models are an approximation of reality. So you're really trying to capture what is the essence of the system that I'm trying to model to answer the question that I want to answer. And with that, you might be abstracting away a lot of information that is relevant in other contexts. For instance, if I'm creating a between host model, I might not care about the exact dynamics of the immune response within each individual. I just say, after 10 days, most individuals are recovered from the disease. And of course, for a between host model where you care about who infects whom and how long does it take for people to infect each other, this is all you need. But if you care more about the exact dynamics of the infection within a single person, when are they going to feel most ill, how will they respond to certain drugs, and so on, you actually need to model that immune system in more detail. So I think this is what George Box here means when he says all models are wrong, but some are useful. All models are an approximation of reality. But if you do it properly, if you approximate the relevant aspects of reality, there might be certain questions that you can answer with your models. So with that, I come to what did we actually use mathematical modeling for during the COVID-19 pandemic. And perhaps some of you are familiar with the work of the Swiss National COVID-19 Science Task Force. These are people who are independent scientists, actually, from different universities all around Switzerland, but came together and, I think it was largely ehrenamtlich, so they didn't get paid for it, but they basically tried to synthesize what is the scientific knowledge that we have about COVID-19 and how one could respond to it. So they didn't say you have to respond to it in this way or that way, but basically just summarized the scientific evidence for certain courses of action during the pandemic. And this science task force also had a large sort of modeling team that I was either directly or indirectly involved with. And some of the questions that they touched on during this period was, for instance, to try to predict the spatio-temporal dynamics of the disease. So essentially, to first of all monitor who is getting infected where and when. So where in Switzerland, in which canton, in which city, and how many infections are occurring through time each day. But then also to use this information to predict where do we think the burden is going to be highest in the next 1 or 2 weeks. And related to this, also to predict where are we going to need hospital capacity? How many people do we expect to become so sick that they end up in the ICU? And this relates, first of all, who is getting infected and where, but also who is most likely to develop severe disease, for instance, because they have comorbidities or because they're older and things like that. Then another set of policy questions were on assessing the impact of interventions, be it mask wearing, closing borders, stay-at-home orders, and all of these non-pharmaceutical interventions. Designing optimal vaccination strategies. You might imagine that there's different optima depending on whether you're trying to prevent deaths or hospitalizations, in which case you want to vaccinate your at-risk populations earlier, versus trying to minimize the number of infections, in which case you might want to target people who are most mobile in the population, for instance, and have most contacts to other people and most opportunities to transmit an infection. And then there were other questions like quarantine duration, and the impact of variants with different transmission dynamics. For instance, do they transmit earlier during an infection? Do they transmit to more people in general? Do they transmit to people that have been vaccinated before or have some kind of immune escape, and so on? So for the remaining let's say half an hour, 20 minutes, I want to highlight a few specific projects that I've been working on within this scope over the past 2 years, primarily on estimating the effective reproductive number of SARS-CoV-2 in Switzerland. So when we're monitoring an infectious disease epidemic, there's a few key variables that are important to keep track of. First of all, we care about the disease prevalence, so how many people are currently sick with the disease, or perhaps what's the occupancy of ICU beds. Then there's the disease incidence, so how many new infections are occurring each day or each week, for instance, how many people are newly reported to be sick with COVID-19, as well as changes to the incidence. So are ever more people getting infected with the disease, or is the pandemic actually decreasing in size? And it's precisely this last quantity that we try to describe using the effective reproductive number, which describes the average number of secondary infections caused by a primary infected individual. And this can change through time, depending on interventions, for instance, simply how many people a person is in contact with on a given day. And it's a very intuitive number, because if this is above 1, each infected individual will infect more people, so the pandemic will increase, whereas if RE is below 1, the pandemic actually shrinks in size. And we've developed sort of both a method to estimate this effective reproductive number, as well as a dashboard which displays these results. These results have been directly used to inform public health policy in Switzerland over these past 2 years. It's also the method that's behind the values that you'll see on the COVID-19 dashboard of the FOPH, for instance. And we even had this kind of infamous moment in December 2020, where the opening or closing of certain establishments in certain cantons was directly tied to the values that we published on our dashboard. Interestingly, the government didn't tell us beforehand that they were going to write our dashboard into the law. So initially it crashed immediately that afternoon when everyone wanted to figure out what the RE values were. Luckily, we got it back up within a few hours. But that's just a side note as to how some of the discussions sometimes went with actual policymakers. More recently, our dashboard was also used when first presenting Omicron to the public, because interestingly, outside of Switzerland, sort of the main country that's been looking at our dashboard and at our values is South Africa, because very early on we had a good exchange with this professor, Tulio de Oliveira, from South Africa, to include province-specific RE values onto our dashboard, and they've been using those since. We also show results for 168 other countries, just using sort of publicly available data and feeding that into our pipeline. So what does our pipeline really do? Essentially, we take observed case incidents, or actually different proxies related to the infection incidence, that be it observed confirmed cases, deaths, newly occurring deaths of people dying due to COVID-19, and new hospital admissions. We take these different types of case incidents and relate them to estimates of the effective reproductive number through time. And really the core idea here that makes this all possible is that the observed incidence of infections contains information on the effective reproductive number, because it describes sort of how many individuals a single infected individual is going to infect over the course of their infection. And perhaps already here, imagine these two observed infection incidences through time. You can already intuit that sort of the scenario on the right has a higher RE. So we're observing, or each infected individual is infecting more people than in the scenario on the left, where we start with one infection, and the next day we still have one infection, and we sort of have one infection until infinity. And indeed, that's the case in the scenarios here that I drew. RE is 1 on the left and RE is 2 on the right. And we made the assumption that each infected individual will infect all of the people they're ever going to infect on the next day. But this is not necessarily the case for every virus or every disease. It's actually probably not the case that you would always infect or be most infectious the day after you get infected yourself. Instead, this might follow some kind of infectivity profile. So you will infect people over multiple days after you first got infected yourself. For SARS-CoV-2, it's actually around 4 or 5 days or so, over which, or 4 or 5 days after you get infected yourself, you're going to be most infectious. And that means that there's some kind of inherent sort of smearing out in the infections already, even with the same effective reproductive number, so the same RE. If you compare this curve here, where all new infections take place over 2 days, we see that it takes a bit longer, it's a bit slower than in this particular case, where the RE is the same, but the infections are just occurring faster. And that's also the big difference here between the RE and for instance, just estimating a growth rate of the infections directly. So with this information, we actually have everything that we need to estimate the effective reproductive number from some observed incidents. So essentially, we have a model that describes how the observed incidence on a particular day depends on the past incidence, as well as the RE on that particular day and the infectivity profile of the disease. And with this model, we can sort of estimate, or we can describe how likely it is to observe the data that we did observe for a particular value of RE, and then use that to estimate the most likely RE that has likely led to the infection incidence that we truly observe in this case. I'm happy to answer some questions about this in the end. This is probably too much detail, but the core concepts to get from this is essentially that the infection incidence contains information about RE, and we can use that to estimate this RE. But we don't normally observe the infections directly, because it takes a while for people to start showing symptoms and to go and get tested, or perhaps even to get hospitalized with the disease or die due to the disease. And this delay means that we cannot observe the infections directly, and we kind of need to subtract this delay from the cases that we observe to get back to the infection incidence and actually truly say something about RE. So the first step in our pipeline is to do precisely that. We use a mathematical trick called a deconvolution to subtract the delay of these different types of case incidents and get an estimate for the infection incidence. And as you can see here, this estimate of the infection incidence, that's the solid blue curve here, sort of precedes the observed case incidence that resulted from those infections. And then once we have that, we can use this procedure that I just described, this epi-estim method, to estimate RE from the infection incidence. But now this estimate does not properly account for all of the variability, basically the uncertainty that occurs between infection and case observation. So that is, for instance, the fact that we don't observe equal amounts of cases on the weekends as on weekdays because people might wait to get tested, for instance. These are all uncertainties that come in the reporting and that are not taken into account in the estimates of uncertainty from epi-estim directly. So that's why we bootstrap the case incidence. Again, this is a mathematical procedure, which essentially means you're drawing different versions of the observed case incidence that somehow seem plausible, like they could have been the true observed case incidence. We estimate for each of those alternative realities, essentially, we estimate the effective reproductive number, and then we summarize across all of these options and get confidence intervals for our RE out. So with that, we now have an idea of our pipeline. Essentially, we take case incidence, different proxies for infections, and estimate the effective reproductive number from it. And why do we take different proxies for infections? Why not just say, okay, we want one estimate for RE from the confirmed cases, which seem to relate to the infections most directly somehow? Well, each of these observations, so these case incidence traces, has different types of uncertainty associated with them. So for instance, the confirmed cases might be biased due to underreporting. If it's hard to get tests during a period of high incidence, maybe less people get tested overall, and we get a biased picture of the underlying infection incidence. And in those cases, you might say people who need to go to the hospital will go to the hospital regardless of whether there is testing capacity. And so in that case, the hospitalizations might be a less biased representation, basically, of the underlying infection incidence. Of course, only a fraction of the infections will end up being hospitalized in the first place. But if this fraction is always the same, our RE estimates will not be biased, because we're always looking at relative differences within just one type of case observation. However, all of these proxies are still somehow collected within the healthcare system. So if our complete healthcare system breaks down, there's triaging in the hospitals, there's no testing of people anymore when they've deceased to figure out their cause of death and so on, all of them might end up being biased. So in that case, we would like some independent proxy which is collected outside of the healthcare system. And this is exactly where the wastewater data comes in. Because already quite early on in the pandemic, typically environmental scientists essentially showed that you can find viral RNA in the wastewater, and that if you quantify this viral RNA every day, for instance, at the wastewater treatment plant in Zurich, Werthölzli, then these measurements follow the trajectory of the case incidence, essentially. And that's what you see here on the right. So the new cases, the case incidence is shown in blue. and the measured viral loads are shown in orange and red. And we really see quite a good match. And the reason is that each person who's infected with SARS-CoV-2 will shed deactivated viral particles into the wastewater when they go to the toilet, when they brush their teeth, and so on. Yeah, essentially this really sort of maps how many people are infected within your catchment area, so the area that's feeding into the wastewater treatment plant, what you're observing in terms of viral load in that catchment. And then to incorporate this now into our pipeline, the big question is, how do we relate these measurements of wastewater viral loads back to when the infections occurred? And so instead of a delay distribution like with the other types of information, where each infection corresponds to an observation, let's say 5 days later or 10 days later or so, and there's a delay distribution of infections to case observation. Now we have individual level shedding load distribution. So there's a function that describes sort of how much virus you're going to shed into the wastewater in days after you've first been infected. And this is all we need, essentially, to then do the deconvolution again and get an estimate of the infection incidence. And this works, and we've been monitoring this for 6 different locations in Switzerland now over basically the past year or so, and we might extend it to even more wastewater treatment plants and also to other viruses that can be captured within the wastewater. I see there's some point in the chat. Okay. So that was the first 2 topics that I wanted to show you. And now very briefly, also the work that we've been doing on variants, because as you probably all know, over the past year or so, the dynamics of SARS-CoV-2 have really been dominated by a succession of different variants. There was Alpha, Delta was big, now Omicron. And we know about all of these dynamics, these differences within the epidemic because of sequencing, essentially, and genomics. So we can get information about variations within the virus sequence that allow us to say, okay, this is now a new variant that might have also different characteristics in how they are transmitted and how they then affect the further dynamics of the pandemic. And the WHO defines a variant as a variant of concern if it exhibits one of 3 characteristics. Either it has increased transmission, it needs maybe a smaller infectious dose to infect a new person, or it has higher viral load and therefore can be transmitted at a better rate, or perhaps it is transmitted earlier on during the course of infection, so when people don't have symptoms yet and don't start changing their behavior. The second thing is if it has increased severity, so if people get more sick due to it, which again might also change the burden on hospitals and so on, or if there's a decreased effectiveness of diagnostics, therapeutics, and vaccines. And this is actually related to the other 2 categories as well, because if you cannot detect a virus, the person who's infected with it will not change their behavior unless they're very, very sick, but they might simply not know that they're infected and increase the transmission. Also, if the virus is has immune escape, for instance, it might infect more people as it does so. And if it can escape therapeutics, it might lead to more severe disease in more people simply because we cannot treat it properly. Yeah, so it's generally interesting and important to know which variants are around, what their characteristics are they of concern or not, and also how they spread and how different variants might exhibit different transmission dynamics. And so this is one question that really came up at the end of 2020. Essentially, we started hearing some news from the UK about a new variant popping up and really driving increased transmission across different areas of the UK, primarily also England. And then we asked ourselves the question, does this new variant have a transmission advantage, yes or no? And is this going to have an effect on the upcoming developments, basically, of the Swiss epidemic? And so this is primarily work from some of my colleagues, Charan Chen, who I think is also taking the course, and Sarah Nadeau, who've been sequencing or sort of leading the sequencing efforts of a lot of SARS-CoV-2 in Switzerland. And with this sequencing, they could determine the proportion of cases at any given time that were due to this particular variant. And you might get a table like this on the left, where you have the total number of cases that were sequenced and which fraction of those belong to a particular variant. And then you can plot this through time, saying, okay, the fraction of all sequences that we have that are due to a particular variant is increasing through time. And by fitting some models to this data, we can estimate the fitness advantage of this particular variant compared to all other variants that were around. Because if it wouldn't have a transmission fitness advantage, it wouldn't increase its proportion. It would sort of stay the same at whatever level it was introduced at. And we essentially found this fitness advantage of something between 40 and 50%, which also fit with some of the results that were coming from other countries. And then if you have these numbers of cases due to a particular variant through time, you can also estimate variant-specific REs. And we found that essentially for non-B117, so non-alpha cases, the RE was below 1. So in principle, the epidemic would be decreasing in size, whereas for alpha, it was still above 1. And this would ultimately drive the epidemic again to increase in the future, unless one would do something about it and increase measures. And if I remember correctly, this is then exactly what the politicians in Switzerland also did, to sort of increase restrictions and to prevent this sort of exponential increase of the epidemic to occur. So to conclude, I've showed you different ways in which mathematical modeling informed the public health response to SARS-CoV-2 in Switzerland, specifically how we developed this pipeline to estimate the effective reproductive number, and what we can do with that to say something about variants, for instance. If you're interested in these topics in general, I can really recommend this course at ETH Zurich, which is organized by some of my advisors, on infectious disease dynamics, where a lot of these sort of mathematical models are discussed in more detail. Yeah, and also the paper that I submitted as recommended reading for this course. And yeah, this is the team. I will leave it to you to check out the different dashboards that we have and so on our estimates. And we'll take some questions now.
Well, thank you very much, Dr. Jana Huisman, for the wonderful presentation and for the brief introduction to mathematical modeling and the work that has been ongoing and has been done in the past 2 years. My name is Charles. I'm the moderator for today's Q&A session. The Q&A session is open now. So if you have any question to Dr. Jana Huisman, please feel free to unmute yourself and ask your question directly to her. Or if you like, you can even use the chat and send your question via chat, and I will read it out to Dr. Huisman. So in the meantime, while people are reflecting your presentation, I do have a question. So Switzerland has basically lifted all its restrictions, right? Due to the decreasing number of patients in the ICU. So there is no quarantine, there is no isolation. People go to work, they get infected by Omicron. And so basically, if there's no restriction, there is well, less incentive to get clinically tested, right? So my question to you is, when large-scale clinical testing is not anymore performed or in a very small way, what is the value of wastewater monitoring?
Yes, it definitely is more useful in those cases, I would say. So I think wastewater has the advantage that it's a relatively cheap way of covering the entire population and really getting a feeling for who might be infected in that population. And it doesn't require any health-seeking behavior, as you already mentioned. So in that sense, we would expect at this point in time the wastewater signal basically to be very similar to before, or at least a similar kind of representation of the underlying dynamics, whereas perhaps as you were saying, the confirmed cases or things like that might be more biased at the moment. So that's also part of the usefulness, I guess, of having multiple of these traces and in comparing the wastewater against the confirmed cases to see whether we might be missing something, whether we might be biased if we would only look at the confirmed cases. Yeah, for sure. But I think, as far as I know, at the moment, testing is still free, or it's still available. So people still do it. I guess we're just missing perhaps people who would have otherwise been contacted by contact tracers or things like that. Sort of, we're missing a larger part of these asymptomatic people or those with relatively minor disease or things like that. But I think quite a few people who yeah, have like proper symptoms of SARS-CoV-2 are still getting tested at the moment.
Thank you. So are there any questions from the panel?
I have a question because you had mentioned how you obviously try to control for all these different variables and anything that could occur in a parallel universe. And then I was wondering, you know, because you mentioned obviously having had this experience, you really tried to control for these things, but was there anything that you felt you were surprised by that you didn't necessarily control for and then came to find that actually it was a significant thing to control for, or something that kind of, yeah, that you just found surprising in your research?
Yeah, that's a good and difficult question, let's say. So first of all, I think in our work, we don't control so much for which variables might be affecting the effective reproductive number. Rather, we're trying to estimate it and show what it looks like, and then allow others to say, okay, what covariates might explain this behavior. So what relationship do we see, for instance, with the stringency of interventions, or does it relate at all to the mobility of the population, like changed behavior in even beyond just what is mandated by the government, but do we see people change things there? And I think we've also tried to do some analyses there ourselves, like I don't know, regression type of analysis, where we're saying, okay, does, for instance, the stringency of interventions predict what the RE will look like at a particular point in time? And we find some relationships there, but we also find that it's generally more complex than just that. And specifically, trying to model human behavior is really difficult, where, for instance, we don't know whether people might just be looking at the number of infections that are currently occurring, like they're reported everywhere in the news and so on, and might be changing their behavior based on that, even before the Swiss government is saying you should be changing your behavior. And yeah, I mean, for instance, one example of that we find is that in Switzerland in general, the RE seems to be lower than you would expect given a particular set of interventions than it is in other countries around us. So it seems like the Swiss are just really listening to the government, or taking their individual responsibility to a greater degree than other countries. And I think that's quite interesting to see and nice to see. And it also illustrates, I think, a bit of the sort of where mathematics can stop. You know, there is still a very large component of sociology and all these things, anthropology, like information that we need about what drives people to behave in a certain way, how they're perceiving measures, how they're perceiving interventions, and so on, that we just cannot capture in our model. But we can see that something is changing, and then say, oh, this might be interesting to figure out what's causing that kind of behavior, essentially.
Yeah, thank you very much. Really interesting to hear your perspective on it, and also to hear that, of course, how things are in Switzerland are somehow encouraging and positive, and great to even know that your what you've published is somehow, and the work you've been doing is also being listened to by scientists and policymakers here in Switzerland as well. So that's even more encouraging, and we are very grateful to have you here sharing this with us. I think there is another question in the chat.
Yeah, exactly. This question is from Silvan Kaufmann. Is it likely that the quality of water analysis will suffer due to the lower testing number? For example, if there is a variant that is not recognized by the monitoring and you miss it?
Okay, I'm not entirely certain I understand the question, but I will try to answer. And then if I didn't capture, please, sort of rephrase and ask again. So the way we do the analysis in the wastewater is that we basically take, I don't know, a tube of wastewater essentially, and sequence everything in it and try to determine which fragments of RNA that we find are SARS-CoV-2, and specifically which variants might they belong to. But we do not specifically target variants that are known. We just target SARS-CoV-2 in general. So in that sense, we should, in principle, always capture whatever SARS-CoV-2 is in there and not be biased essentially by whether there's a variant that we're not aware of. What could, though, change what we're observing in the wastewater is sort of the host internal dynamics. So if a particular variant leads to a lower viral load, so people shed less virus into the wastewater, we're going to observe less virus overall in the wastewater. And initially, we will not know whether that is because there are less people infected or because this is a variant with less shedding. And that's something that we've actually seen with Omicron, for instance, where at least BA1 seems to be shed at a slightly lower level into the wastewater than we were expecting from Delta. And so we were basically getting slightly different results from the wastewater than we were observing with the testing, because in those cases, people continue to get tested, but we saw less in the wastewater. So we kind of knew, okay, this has to be due to less shedding. And so there's some dynamics there where really having these both of these different traces gives us information about what might be going on. There was another component to that question. Yeah, lower testing number. Yeah, so I guess if we have less testing overall within the healthcare system, and we rely solely on the wastewater, then we don't have information to correct that anymore, if there would be a similar kind of case as with Omicron. So that's true. I guess in that case, we would hope that we still get additional information from other types of studies. So there's more and more studies now, kind of sort of prospective cohort studies where they're seeing who's getting they're testing people every day, both sort of nasopharyngeal swabs as well as feces samples. And then depending on who gets COVID or not, you basically get a nice time series of how much they're shedding at a given point in time. And if these kind of studies do continue also for new variants and things like that, we'll always have information sort of on how much shedding we expect. And this can really inform our wastewater data also without having like full coverage of clinical testing everywhere.
Thank you very much. Are there any other questions? Yes, there's a question from Dylan Croft. How much time would it take to figure out if there is less shedding of a viral load? Any viral load? If people stop testing for the virus in question?
Yeah, that's a good question. So essentially, once a new variant becomes dominant or starts to be present in, let's say, more than 30% or so of the cases, I guess you're going to start seeing an effect on the wastewater measurements, that you're getting less virus there. Basically, you're seeing less virus. How long it would take us to then realize that this is because of a new variant if we're not actually testing for new variants, that's tricky. I'm not entirely sure. I think there's fairly good surveillance around for SARS-CoV-2 variants at the moment. So simply people who are scanning all of the sequencing that we're getting and seeing, are there certain mutations that are sort of increasing in frequency, and can we do this kind of analysis? Basically, can we see that there's a certain proportion that is increasing, that suddenly all the virus that we're sequencing looks different than it looked before? And if we have this kind of information that there is a new variant coming up, and simultaneously we see less viral loads in the wastewater, for instance, we might start to wonder, like, okay, could it be that these two things are linked to each other? Could it be that we're now missing cases, essentially, or that there's a larger proportion of cases that this same virus signal is corresponding to. And again, that would then be a signal that could be communicated to the public health authorities to do further testing, further inquiry, essentially, of what might be driving these dynamics.
All right. Yeah, we are running almost out of time. We have come slowly to the end of this Q&A round. I think I will hand it over to Emily. And thank you, Dr. Huisman, for your presentation.
Thank you.
Thank you very, very much indeed. It's been great just to have your expert insights on all of this, and that we were able to ask our questions and to know more about this very relevant topic from someone who's also been able to influence science and policy here in Switzerland. Thank you also to Charles, of course, for bringing us nicely through the discussion time. I'm trying to share my screen. It is not cooperating with me. Let me see, but that's okay because it's nothing too significant. You may have guessed, obviously it is coming up now till the end of our time together before the midterm break. So in fact, we don't have a session coming up next week. So we will only see one another once again in 2 weeks' time. But in the meantime, I wish everybody all the best for the midterm quiz, and also a very, very happy Easter to all of you. And once again, thank you very much to Dr. Huisman for being here and for giving us this excellent lecture. And thank you again to Charles also for leading our discussion time. And with that, I close our session and wish you all a very happy Easter and a nice evening. Bye, everyone.